Hey, wait a minute, man. Hey, check this out, man. Tell it. There was nucleic acid, right? There was nucleic acid, right? He was making his way around the nucleus of this cell, right? Hey, he saw this pretty little chromosome, you know what I'm saying? He stopped, he took a deep breath, he said, if I were an enzyme, I'd be DNA helicase so I could unzip your genes.
Hey, you like that DNA, man? Hey, man, I got like 20 billion kilometers of that DNA, man. Hey, my man on guitar. Hey, fool on the drums. Hey, everybody just crowd around the mic. I'll tell you all about this deoxyribonucleic acid. First, I'm gonna start off like this. Hey, help me sing it, homeboy.
It's the deoxyribonucleic sugar and the phosphate groups. Baby, that's all we need to make up the backbone of DNA before we can replicate. With the nitrogen base, yeah, the nucleotides, purines and pyrimidines, adenine and guanine and thymine too. Don't forget about cytosine. So join your bases, A to T, D to C. With a hydrogen bond, we can move it along and maybe even make protein. So join your bases up, A to T, G to C. Think you know all about your DNA? Well, man, you sure are wrong.
Well, the template strand in DNA will make two daughter strands. The enzyme helicase breaks them up, and then DNA expands. Now all of these bases gotta match up with their complementary base pairs. So G goes with C and A goes with T, cause no base U is there. With hydrogen bonds, they are joined up, helped out by DNA polymerase, and with the phosphate groups, the sugar groups, they're then joined by DNA ligase. Both these enzymes help with building a double helix. Ain't that thrilling? Without a copy to see, there'd be disarray, so the parent strand makes the DNA. Since each pair has half the strand, semi-conservative is what it's called. Do you understand? DNA replication is during interphase. In this stage, it goes without delays. Replication happens fast in the cell. My body can do this so very well. I got a parent strand and a bunch of enzymes, so DNA can replicate one more time.
It's the deoxyribonucleic sugar and the phosphate groups. Baby, that's all we need to make up the backbone of DNA before we can replicate. With the nitrogen base, yeah, the nucleotides, purines and pyrimidines, adenine and guanine and thymine too. Don't forget about cytosine. So join your bases, A to T, D to C. With a hydrogen bond, we can move it along and maybe even make protein. So join your bases up, A to T, G to C. Think you know all about your DNA? Well, man, you sure are wrong.
Before the protein is ever made, first we have to make an mRNA. So the right code can be transferred out of the nucleus, it's varied without a doubt. The DNA is split by helicase. It breaks the bonds between every base. RNA polymerase fixes it up by bringing more nucleotides to join the club. But RNA uses different bases than DNA, that's just what the case is. Cytosine and guanine are just the same, but uracil bond with adenine in thymine's place. Our RNA has been created. Without ribosome, it's slightly tainted. The introns have to be removed. The exons must stay or we'll just be confused. Almost every living cell under the sun goes through this thing, it's called transcription. To turn DNA into a protein, there are more steps that are in between.
It's the deoxyribonucleic sugar and the phosphate groups. Baby, that's all we need to make up the backbone of DNA before we can replicate. With the nitrogen base, yeah, the nucleotides, purines and pyrimidines, adenine and guanine and thymine too. Don't forget about cytosine.
The mRNA is ready to go, so it makes its way to the ribosome. It attaches at the start codon AUG. Initiation has started, man, that's what we see. The anticodon is brought on down by tRNA. Yeah, that's what we found. This tRNA also carries with it a very particular amino acid. Amino acids coincide with just 3 bases. There are still a lot of combinations. Within our bodies, we use just 20. Out of 64, that's not too many. There are, however, only 3 for stop, and UAG will do the job when the peptide strand has grown in size, and after the stop codon has arrived. A release factor cuts off the protein chain. The mRNA is preserved. You can use it again. A polypeptide was formed through translation. That's protein synthesis, protein formation.
It's a deoxyribossugar and the phosphate groups. Baby, that's all we need to make up the backbone of DNA before we can replicate. With the nitrogen base, yeah, the nucleotides, purines and pyrimidines, adenine, guanine, and thymine too. Don't forget about cytosine.
Hey, check this out. Have you ever been trying to make RNA, but something just don't work out? You've got the wrong acid and it ain't no good, and it's making you wanna shout. Addition or deletion, you missed a base, or maybe it was substitution. The sequence of codons, it just ain't right, and it's caused a mutation. Point mutations come in 3 different types. Silent really ain't that bad, but missense and nonsense can cause a shift, or even stop the peptide strand. Caused by radiation, or UV light, or maybe even pesticides. These mutagens can really mess things up. Pray you're not that 1 in 10 billion. So deoxyribossugar and phosphate groups. Baby, that's all we need.
Excuse me.